I don't know why it does that, but it's like it goes all the way to 0 and then comes back up. It just go to half. Okay. All right, guys. So my name is Joshua Horton. We'll get started here. I am the lab coordinator for Bio1102 and 4102. So I'm the guy that does kind of all the background stuff. I may have seen you guys on Thursday periodically. You may have seen me walk in, or I said hi to you all. Mostly I'm doing stuff on Fridays with you guys. Oh, goodness, that's loud. And let's see if I can get this to not peak and do the loud screechy thing. All right, can you guys hear me still okay? Yes? Good, okay. So basically I just handle kind of the Friday stuff, but I also am the guy that takes care of all the media stuff that you guys get and gets things set up so that you guys can have your experiments ready. And I'll peek my head in from time to time and make sure you guys are doing okay and ask you guys questions and generally torment you and make your lives difficult. But not really. I'll try to make your guys' lives less difficult as much as possible. All right, so let's go ahead and get this started. So we're going to be talking about what's going to come up next week, which is microscopy and basic handling and culture techniques. So typically what I do is when I start lab lecture, I will go over something called Cool Science Thing of the Week, which is typically something kind of really new and modern, kind of close to what we're talking about in class. In this case, I just kind of did a fun one that I happen to like, but it's kind of related. And it is the basis for our bonus questions for our lab practicals, so keep that in mind. So if you want to pay attention, you might get some bonus points on your lab practical, which adds up quite a bit sometimes. All right, so this one we're going to talk about is about this guy who's an artist with bacteria. He does some really cool stuff. Here's his website. One of the things he does is he does some Christmas ornaments using luminescent bacteria. And the one that he typically uses for luminescent bacteria is this one called Photobacterium phosphorium, which is a seed-dwelling bacteria. It is very picky, though, on what it grows on. It tends to like to only grow on, it's a very specific type of media, and on top of that, you have to supplement it with seawater, so salt, and then also on top of that, you have to give it 1% glycerol. If you don't give it both of those things, it won't luminesce. So it's very picky, and you have to constantly be adding more of those two chemicals in order for it to stay alive and function properly. Where did that skip? Okay, so keep that in mind. Let me adjust the microphone level real quick to see if I can make it less super loud. Okay. So he's like, okay, let's see if we can kind of find something that's a little bit more robust and maybe won't cause me to have to supplement it constantly, because trying to supplement Christmas ornaments with glycerol doesn't work very well. So he stumbled upon kind of these bacteria. They're called Photobacterium. There's two species that he looked at, which is Asymbiotica and Luminescens. And they actually grow really well. They're normal land-dwelling bacteria. So he just put them on regular old media, and they grew pretty well. They made a nice luminescent glow. And so then what he is trying to figure out is kind of like the optimal growing conditions for them. And so what he did is he added in 1% glycerol and 1% glucose in different concentrations and grew them on the same media. And what actually happened is, and you guys can kind of see it over here, is that it actually reduced in luminosity. So it produced less glow. And on top of this is one in the daylight. One of them actually started producing a secondary dye that nobody had seen before or documented. And the general rule of thumb is for things that like to glow or luminesce, that's a lot of energy intensive excess work. So basically if you feed them more resources, the bacteria will glow more. But in this case, you fed them more resources and the bacteria glowed less, which is really unusual. And so it just goes to show how like small little changes can have really big impacts on different bacteria, especially not even ways that you can interpret, you can like predict. In this case, it's almost always true that if you add more nutrients, it glows better. But in this case, it actually did the opposite and did something even wackier, creating a new little dye that it produced that it doesn't usually produce. Now, so a little bit of background about these bacteria. The Photobacterium are actually really interesting. The way we found them is that they actually grow in these nematodes, which are kind of little see-through worm things, and those nematodes do a variety of things. A lot of times they're associated with things like rot, so they like to grow on like fruit trees a lot, and that's where we find them often. But they're going to do a variety of things. In this case, the species that these ones like to do, they inhabit the guts of these nematodes, and the nematodes infect the guts of caterpillars, specific types. These ones are specific types of moth ones, and they grow in there and make them glow slightly, faintly. Why do they make them glow, you ask? That's a great question. We have no idea. Like, there is zero reason for them to glow. Why would it glow? It doesn't make any sense. We can't find any reason. And because it's infecting these guys, and it is considered an infection for them, it's ideally bad for them to be infected with this glowing bacteria. My crazy thought, and I have no proof to put this back, to put this forward, but this is my suspicion, my conspiracy theory, is that it actually makes them glow so that the caterpillars are more likely to be predated upon by birds or bats or something. So they grab them, eat them, transport the bacteria somewhere else. They come out of the bird, and now they've got a new home to infect new caterpillars with, because if the bird's grabbing caterpillars from one area, chances are they'll find caterpillars in another area, and now the bacteria and the nematodes together will be in a new area with new hosts to infect. Seems like a pretty good idea. That's just my wild suspicion. Now, in terms of humans, though, we actually do know that these bacteria sometimes infect humans, but very rarely. We have two examples of this commonly, which is in World War I and in the Battle of Shiloh for the Civil War. We noticed that there were soldiers who would sometimes get glowing wounds. We called them angel's glow. And the reason why is because those soldiers had a higher chance of survival from their wounds if they have the angel's glow. The reason for this is that we know that the photobacteria and bacteria often produce antimicrobial agents that kill off other bacteria, but they're not really designed or set up to handle our immune systems. They're designed to handle those of caterpillars and nematodes, and not really our immune system. So our immune systems are actually able to get rid of the photobacterium bacteria very easily, or relatively easily. But the bacteria that we normally struggle with, they're able to get rid of rather easily because of their antimicrobial agents. So it actually kind of works out really well for those people. Now, there are some people that will get infected and sometimes do get sick. Typically, they are older, have weakened immune systems. There's actually one example in Texas of an older gentleman who was gardening, and he got a wound on his hand while he was gardening, and it got infected, and they sent it off for testing, and it turned out to be glowing faintly. And so they put his wound in a dark room, and they could also see the glowing as well. And that was one of the few examples of Photobacterium asymbiotica infecting a human. Typically it doesn't, but occasionally we will find people with these kind of glowing infections. So, very interesting little thing. Okay, so that's our cool science thing of the week. Basically, if you take home message, media affects luminescence in many ways. In this case, increased nutrients leads to decreased luminescence for Photobacterium, Photorhabdus species. Okay, one of the other things that we'll typically have on here is a slide about safety, so we'll talk about safety issues for the lab. These are important, so make sure you guys kind of note them down. The first one is about just handling the microscopes. The microscopes are pretty heavy, so you could drop them and break the microscope or hurt yourself, so be careful because they could break a toe. The way you hold the microscopes is always from the back. There's a little handle on the back, and you put your hand on the bottom, so you always carry it with two hands, and you take it back to your seat. The microscopes are assigned by seat number, so if you guys didn't notice before, if you pulled out your drawer from where you sit, the drawer in front of where you sit directly, there's a little number in there. Each of those numbers corresponds to a seat, and that seat number corresponds to your specific microscope. So make sure you guys grab them from the right spot, what have you. Okay, silence phone. All right, the other one. Whenever you handle slides, please be very careful. Slides are made of glass, and glass does shatter, and shattered glass is very sharp and can cut you, so please be careful with them. If you happen to break a slide at any time, make sure you guys let us know. Also, don't leave slides in the microscopes because that can cause a variety of problems, one of which is having them break accidentally, so please be careful of that. Also, whenever you guys use the microscopes, make sure you guys clean them between slides. This isn't necessarily a safety thing, but it is a good habit to make sure the microscopes are in proper working order. So make sure you guys always do that. The last one is to make sure to never use the only use the fine adjustment knob, I should say, on the 40x lens and the 100x lens. Never use the coarse adjustment knob. Coarse adjustment knob makes the stage move up and down in by a high degree, so it move up and down quite a bit. You can accidentally break the glass slides, which is very close to your hand, as well as the lens. The lens is very expensive to replace. So it does pose a safety hazard. So please be careful never to touch the coarse adjustment knob while you're on the 40x or 100x lens. Okay, so the microscope that we'll be using is shaped like this guy. You should know the various parts. I'm not going to go over all the parts, but I'm going to go over a couple parts that tend to trip people up. And the first one is the objective lenses, which are these guys right here. They sit on this thing that's called the rotating nose piece, which is this guy right here. So the rotating nose piece is the thing that turns the lenses to different parts, and they kind of click into place when you're on the right lens position. The other thing is underneath the stage, there's this guy right here, which is the iris annulus diaphragm. That's underneath the stage, and that kind of opens up this little iris, much like the iris in your own eye. So it squeezes open and close like that, and it allows light to come in a different kind of angles so that it's a little bit easier to visualize the edges of some things. So you might need to make some adjustments using that. Also, lastly on here is your adjustment knob. So you've got the coarse adjustment knob, which is the big one on the outside, and the fine adjustment knob, which is the smaller one on the inside there. You'll be doing things with microscopes both days, both Tuesday and Thursday for you guys. On day one, you guys will be looking at blood smears. The goal is to familiarize yourselves with basically getting things into focus, going up to the maximum magnification, moving around on the stage, and kind of being able to find different things on there. Day two, we will be mastering these skills, so really refining them. Blood is generally pretty easy because blood cells are, human cells in general are much larger than bacteria. Bacteria are much smaller. So we'll do some gram-stained bacteria that are pre-made for you guys, as well as a slide that you guys will make based off of your culturing techniques. This is the second lab that we'll also be doing overlapping this one. And we'll talk more about that in a second. Okay, so what will you be doing? Whenever you sit down to use your microscope, first thing you want to do is an ocular adjustment. The oculars of the microscopes adjust and you need to adjust them to fit your personal interpupillary distance, which is a really fancy way of saying how far apart your eye holes are. That's basically all it is. Everybody has different spacing, and you have to readjust each time you sit down at the microscopes because it changes from person to person, and because these are shared between sections, you'll always kind of have to readjust it. What you'll want to see is if you have two separate kind of images like this, they're kind of stuck together, that means that they are just not aligned well. So you want to bring them together or pull them apart just enough until you see one nice large image, so a big field of view. The other thing that can affect this is how far away your eyes are from the oculars. You don't have to have your eyes right on the oculars, but they have to be pretty close, about 1 centimeter away. And it takes a little bit of practice so that you can kind of get your head dead set. So you kind of want your eyes straight ahead. If you're a little to the side or a little to the up and down, it will not let you see a clear field of view. So if you're not seeing a big field of view where you can see kind of everything, then you're probably not in a good position for your head, or the oculars aren't quite dead set for your eyes. So you'll have to play around with it till you kind of get a good field of view. Focusing a microscope. So there is actually a video on Lab Archives that you guys can view that kind of walks you through all these steps and shows you what you should see on a slide. But I'm going to kind of go over this with you guys as well. And your TA will also show a demonstration in the lab on the microscope for the instructor's desk. What you'll do is you will start always at the 10x or the 4x. You don't have to start on the 4x, but some people like that extra safety, so you can start on the 4x as well. It's the same process for both. When you do this, you want to make sure that the stage is all the way down at the bottom before you do anything else. So put it all the way down at the bottom, click it into the 4x. You will then take your slide, you will stick it in there and clip it into the stage, and then you will scroll away from yourself to bring the stage all the way up to the highest position it can be in. And then you want to look through the oculars and scroll back towards yourself, which will lower the stage slowly using the coarse adjustment knobs, until you see something flash into focus on the slide. Now, when you do this, there's a little bit of kind of a trick to be able to predict what you want to look at. If you look through the slide, you will see light shining through it. And so wherever the light's shining through is what you'll see on the microscope. So if you position the slide on something that's if you're looking at bacteria stains tend to have these little purple streaks in them. You can put those purple streaks so that you know that you're going to be looking at something when you actually look through it on the microscope. Otherwise, you're kind of leaving things to chance. So basically, make sure you're looking in a good spot before you start. When you start looking through the oculars, you will use the coarse adjustment knob and scroll down, because it's going to be at the very height, the very top. You'll scroll down, and you're going to bring the stage down just a little bit, and you're going to look for something to flash into focus, something that you want to focus on. Once you see something kind of get mostly in focus, you're going to use the fine adjustment knobs, which are the smaller knobs outside of the coarse adjustment ones, and use those to go up or down until it becomes a sharply focused image. Now, bacteria are actually a lot more like you than you think. They are also three-dimensional, so they have height to them. In the example over here, these guys are actually called cyanobacterium. They're very large three-dimensional structures. And at the top, that first image, the cyanobacterium is right here, and the microscope is actually in focus right above it. So it's all kind of hazy, and you can't really see that much. The second image is where the microscope plane is actually in the middle of the cyanobacteria. So it's actually a mostly pretty good in-focus image. The last image is actually focusing through the bottom of the cyanobacterium. And so you can see how there's a bunch of edges to it that are really, really fuzzy and not in focus, but certain parts of it are in sharp focus. This one gives you kind of the best field of view, and you can have to make this decision for yourself what you want to look at, but just be aware that these bacteria are three-dimensional, so there's going to be kind of like a sweet spot where it's just good enough to give you all the information that you want, and that's what you're trying to find when you're on the microscope. Now, when you do this though, keep in mind, when we go into the next power, which in this case will be the 40x objective, so you'll rotate the nose piece over. You're not going to touch the stage at all while you do this. You're just going to click it over in place, and because it's parafocal, it means that you will jump right roughly to the correct focus when you go up to the 40x lens. Now, that's only if you're dead in center. If you were like a little bit above or a little bit below what you were looking at, what will end up happening is you will end up out of focus. And that's okay, because we're just going to use the fine adjustment knobs to bring us back into sharp focus. You're not that far away. So you'll turn the fine adjustment knob a quarter turn forwards and a quarter turn backwards. Whichever way is better, keep going in that direction. If neither is better, you can go up to a half turn or all the way up to a full turn. And if you still can't get it, you want to go back to 10x or 4x and try it again, or raise your hand and ask your instructor for help. This is actually the best day to ask for help in this, because eventually we're not going to be helping guys focus microscopes anymore. You guys will be all on your own, so make sure you guys ask for help if you need it on this day. Now, if you noticed, I talked about the 40x and the 10x lens, but over here I've got 100x and 400x. That's because this is the total magnification that you're looking through. You're looking through the magnification of your objective lenses that are on the rotating nose piece, but you also have a power 10 objectives for your oculars, so they also have a magnification. So you multiply them together, and it becomes a total magnification of 100x and 400x. Now we're going to go up to the last one, which is the 100x lens, so 1000x total magnification, and you're going to run into the same kind of issue here, except it's going to be a narrower range when you jump up to 1000x. So if you're off just a little bit on the 400x, you're going to be way off on the 1000x, so just keep that in mind. But before you click it over to the 1000x, you need to actually add a drop of this stuff called immersion oil. And the reason why is because oil helps direct all the light so that it goes straight up through to the lens. Otherwise, light kind of scatters, and you have a really hard time bringing everything into sharp focus. So what we do is we click between the 40x and the 100x lens. We'll add a drop of immersion oil onto the slide, click the lens over to the 100x lens, and let it sit there for a second, and then bring it into sharp focus with the fine adjustment knobs. If you aren't able to bring it into sharp focus, there's a couple of things that are the most common reasons why you can't do it in 1000x. The first one is you forgot to do immersion oil. Without immersion oil, you'll never get into sharp focus, so keep that in mind. The second thing is if you have the slide upside down. So you want to have the bacteria on the top of the slide. If it's upside down, you'll actually be able to bring it into focus on the 10x and the 40x, but not the 100x lens. And the reason why is because the 100x lens has to be really, really close to the sample to work properly. And the thickness of the glass is actually so much that the bacteria down here, you can't bring it into focus. So if you can't bring it into focus at all, just double check quickly that your slide isn't upside down. That's the most common issue. Okay, so you'll snap it over to the 1000x, you'll bring it into the fine adjustment knob to bring it into sharp focus, and then, what the heck? Okay, did it freak out on the screen for you guys? Yeah, it did. Wow. Okay, that was weird. The next thing you'll do is once you get it in, you'll look around and you guys will take a picture. Now, there is a small issue with more people getting newer phones that have multi-lens cameras. This is actually a real phone, by the way, like a real camera, like with all these lenses. That I thought it was made up, but nope, that's actually a real camera. When you try to take a picture with the microscope with a multi-lens camera, it doesn't work very well. But there are some tricks to fixing it. The first thing you want to do is set your phone to portrait mode. and then you want to set it to 1x or 2x zoom, and that way it'll tend to snap it down to just one lens, and then you'll be able to take a good picture. Otherwise, it just doesn't work very well, so just keep that in mind. And if that doesn't work, definitely let us know. We're still trying to kind of adapt to this, and because there's so many different phone types, you might run into different kinds of issues, but that seems to work for the majority of people so far right now. So put it into portrait mode. The other thing that happens, just like with your head, if you're a little off to the side or up and down or too far away, it doesn't take a very good picture. So you want to kind of get it pretty close to the oculars, but not touching it, because then it doesn't work at all. So about 1 centimeter away. And it helps if you hold your hand on the side of the ocular lens to kind of hold your phone steady so you're not moving around this way and that way. And the other thing that people have found really helpful is to set it to burst mode, so you can just hold it down and take a couple of photos, and choosing which photo is the best, it's kind of the most in center. Some people tend to have a lot of difficulty doing that, but once you kind of get it down, then you don't even have to do that. You can just do it with a single shot. But that tends to help out some people. I have very shaky hands, so that's one of the things that I use as well. But eventually, I've gotten good enough where I can just take one quick photo and get a good shot. Okay, so picture, there you go. That's how you take a picture. Now, with the first thing that we're going to be looking at, and sorry to flash bang you guys with a bright screen again. For human blood, there's a couple of things you're going to be looking at. The first one, it's the easiest thing to find in all of this, I don't even have a great picture of this, is just red blood cells. They should look... I have, sorry guys, no idea why it's doing that. Okay, hold on. I'm going to mute this for a second. Okay, I think that fixed it. I'm sorry. This was unscrewed. Okay, hopefully we won't deafen you guys anymore. Okay, red blood cells. They should look like nice little pink donuts, because they kind of have a hole in the center. There's not really a hole in the center, it's just see-through. And that's the thing you'll see the most of. Now, if you are looking at the slide and you see a big glob of stuff kind of webbed together, that is not a good spot to be looking in. That's where the blood cells are too thick, because blood smears are literally smears of blood on a slide. One area where the smear starts will tend to be globby like that and not look very good. You don't want to look there. And if you look and you see a bunch of shredded up red blood cells, that's the other end, and that tends to happen as well once you get to the end of the smear. So you want to kind of find a nice middle area, the Goldilocks zone for this one as well, where they look like red blood cells. So if they don't look like red blood cells, you want to look somewhere else on that slide. The other thing that you want to look for is white blood cells. White blood cells have nucleuses, and that's where they have these dark little purple bits, because those are nucleic acid stains that give you a good visualization. Red blood cells do not have nucleuses. They're actually one of the few cell types in humans that do not have DNA in them or nucleuses. They're just there chilling out, pre-programmed cells. They're kind of cool like that. So white blood cells, there's a couple that you have to be able to identify. They're really easy to identify. The first one that's most common is neutrophils. And these guys tend to have these things that they're called multilobate nucleuses. So to me, it kind of looks like dumbbells chained together. But basically, it's just a bunch of lobes together with a bunch of cytoplasm around it. Cytoplasm is the clear stuff that is around the nucleus, the dark purple stuff. The other one that's pretty common that you guys will see is a monocyte. Monocytes are these kind of bean-shaped nucleuses, and they have a whole bunch of cytoplasm around them. Now, the last one that people tend to confuse with monocytes are lymphocytes. Lymphocytes have a large circular nucleus and a tiny little crescent of cytoplasm. So you can see that even though these both have really large nucleuses, this one just has kind of a tiny crescent, and it's got a very round nucleus, not a bean-shaped one. The last two here are basophils and eosinophils, and they're actually kind of unusual in that they represent a very small percentage of blood. I think they're like 0.4% and 0.1% of all blood. So there's not a lot of them just floating around in blood free. So you might have trouble finding them. You'll find probably at least one of them. But if you don't find both of them, don't freak out. That's pretty common. They are very hard to find, much like Carmen Sandiego. I don't know if you guys know who that is. Famous cartoon character that was a criminal that was hard to find when I was a kid. Okay. Basophils are purple. They are both basophils and eosinophils have purple nucleuses that are bilobate, so two lobes. But basophils have gritty purple granules in them, and they like to take up basic stains. So that's why they're called basophils. Basic stains, basophils. There you go. Eosinophils, on the other hand, have these pink gritty granules there, and they are called eosinophils because they like to take up eosine dye, which is kind of this bright reddish color, pinkish color, and so they really like to take up that type of dye, so they're called eosinophils. They like to take up eosin. They love eosinophil. Okay, so that's how that works there. That's the name. So you'll probably see one of these two, but probably not both, and they're kind of hard to find. So they're much lower in population than lymphocytes, monocytes, or neutrophils. Okay, on day 2, you guys will have an opportunity to look at Gram stain smears. These ones, unlike the blood, you can kind of get away with looking at the blood at the 40x. Really, you want to go to 1000x, but because blood cells are so big, you can probably still see them pretty well at 40x, on the 40x lens. But these guys really, you need to look at 1000x under oil immersion. And the reason why is because they are much, much smaller. So we'll give you guys a variety of cells to look at. So Micrococcus luteus, Bacillus subtilis, Serratia marcescens, and you can just kind of play around and look at those ones. Now, the other lab that we'll be doing in conjunction with this one that will be tied together is basic handling and culture transfer techniques. I'll just make sure I'm doing good on time. Great. So this one's basically showing you guys how to inoculate stuff, which is kind of the foundation of what we're going to be doing the rest of the semester. So keep in mind, this is pretty important. First things first, if anything spills or breaks, make sure you guys let your instructor know immediately, and we'll clean it up together. By far, the worst thing you could do in this lab isn't spilling or breaking something. This is a teaching lab. I expect accidents to occasionally happen. That's totally fine. We have procedures set up to handle this. The worst thing you could do in this lab is spill something or break something, not tell us, and then clean it up wrong, and then have a contamination issue every single section after that, and maybe for the rest of the week. There's really easy, simple cleaning procedures. Nobody will get mad at you if you break something. Nobody will yell at you. You won't get in trouble. We'll just work together to clean it up, and we'll take care of it. That's the best way to do it. So never, ever try to hide something if something breaks or spills. Just let us know, raise your hand. We're used to it, and it's not the end of the world by any means. So just let us know. The worst thing you could do is just try to hide it and just have a contamination issue every section afterwards. Which is also the reason why, in case you guys were noticing, that we stress highly to make sure you guys wash your hands at the beginning and end of lab, as well as decontaminating your working area, because that helps. We decontaminate our working area at the beginning of lab and at the end of lab, because you never know. Maybe somebody spilled something before you got there, and now there's a big contamination issue, and this gives you an extra layer of security and safety. The organisms we are going to be using are BSL-1. That means that they typically don't cause disease in humans, but we are going to be treating them as though they were BSL-2 organisms. And the reason why is we're just getting in the habit of doing that. We treat every organism in lab as though they were BSL-2, just to be on the safe side and create a standard of behavior. Once we inoculate our items, we're going to make sure that they go into the proper incubator. Your lab instructor, your lab TA will tell you which one they go into. Anytime we have a stock culture, that's a culture that we give you guys to inoculate your media with. You guys always want to dispose of it in the proper way, and your guys' TA will tell you guys what to do for that. Lastly, you'll always, of course, want to work with disinfecting your working area prior to starting anything and after you get done working on everything. And lastly, you want to wash your hands before and after you are done with the lab. We always wash our hands before lab because we just don't want to bring any bacteria from our outside environment to our working environment to cause contamination. And we don't want to bring any bacteria from our working environment to our outside environment after we get done with the lab. That's why we do it before and after. Okay, so basic handling techniques. Day 1, we're just going to start by doing some foundational inoculations and making cultures. Those cultures we're going to use on the next day. So on Thursday, you guys will then start using micropipetters. Maybe you guys have used them already a little bit. We'll use them quite extensively in this lab. And you'll use the micropipetter to practice pipetting, but also to use a wet mount slide, which we'll use from the bacteria that we made in day 1. Okay. So the process of transferring organisms from one media to another using aseptic techniques is called subculturing. Why do we do subculturing? Well, sometimes we want to make bacteria grow in an active growing state. Sometimes we want to isolate individual colonies, so there's a lot of reasons why we might want to subculture. There are a couple of rules for transferring bacteria in terms of what instrument you use. Typically in your center drawers, you have this thing called a loop and a needle. A loop, as you might be able to imply, has a loop on the end, and the needle looks kind of like a little needle. It's basically just a piece of wire that sticks out straight. You always choose the tool that works the best for your needs and does the least amount of damage to the media that you're going into. So for example, you could use a needle to go from a slant or broth into a deep. In a deep, you have to stab the needle in and pull it straight out to create a smooth growing pattern. If you don't use a needle, then if you use a loop, it would gouge the auger a bunch and ruin everything. If you were going into a streak plate, you'd always want to use an inoculating loop because the loop will create a nice even streak on the plate where the needle will gouge and cut into the auger, and you will get a bad result when you do that. For something like a broth though, it actually doesn't matter. You can't hurt a broth. So a loop or a needle, doesn't matter, whatever for a broth. The last one that's actually not on here is the slant. Slants can be both a loop or a needle. It just depends on what your intended purpose is. So if you're trying to grow just a bunch of bacteria, a loop's fine. If you want to try to identify cultural growing technique or characteristics, you'd use a needle because it makes a nice thin line. Okay, so first one up we'll talk about is tube inoculation technique. I know that there's a bunch of lists right here, but there's a part 0, which is label everything, and I'll point out why this is really important, and why it's important not just for science, but also for nursing as well. By far, the worst thing I've ever done in labs has been to make a bunch of stuff and not label it well, or not label it at all. And I'll go in my brain and I'll be like, yeah, no, I can definitely remember what this obscure tube is. And then like 2 days later, a week later, a month later, I'll look at it and see some cryptic, weird labeling scheme I put on there, or didn't label it at all, and I'll have no idea what it is. It could be something dangerous, it could be something innocuous, it could be the thing that I'm looking for, or it could be something completely different. Always make sure you guys label, but the most important thing on here goes with this. Never label caps. So we never label caps on test tubes because we reuse the test tube caps, and when you draw on caps, it kind of gets locked in when we autoclave to sterilize them. So always make sure to never label caps, but label on the tube itself. Tubes are disposable, caps are not. Okay, so first things first is you guys will label it. Typically the way you label is something that's easy for you to identify, the name of the bacteria, your name, and the date. Those are kind of the bare minimum requirements. And you always do it with Sharpie on the test tube. Okay, so how do you start off with this? Well, you'll take your inoculating tool, loop or needle, and you will sterilize it in this thing called the Bacti-cinerator. If you guys looked on your desks and you saw this kind of beige thing with a metal tube cylinder on top of it, that would be the Bacti-cinerator. Those take a while to warm up, so whenever you have to inoculate anything in class, you want to turn them on and let them heat up for about 5 minutes. It takes 5 to 10 minutes to work, so keep that in mind. Once they're hot on the inside, so they kind of glow orange on the inside, you'll take your loop or your needle, you'll stick it in that hole and let it sit there for about a second, and then you'll take it out. It's actually like 400 degrees Celsius. It's like way hotter than it needs to be to sterilize, so it's very, very good and very quick at doing this. So just stick it in there for a second, take it out, and let it cool down, and then it is considered sterile. Most important thing though with that, do not take your loop or needle, stick it in there, and just leave it in there hanging, because it will actually melt the metal and snap off inside there, and then I'll have to take it apart, or it will ruin the coil that's on the inside of there, and it's no fun for me, so please don't do that. So just stick it in there for a second, take it out, you're good to go. You will then, and I think I forgot the thing that I was going to bring. We're going to use these for demonstration purposes. Your test tubes, pretend these are test tubes. You'll take them and put them in your non-dominant hand. So I'm right-handed, I will put them in my left hand, and you'll put them in the kind of this V-shaped pattern like this. Your TA will show you an example of this in lab, but I'm going to show you guys like this right now, just real quick. You will put them in your non-dominant hand and hold them at an angle like this. You will take your loop and needle and you'll hopefully hold it like this, just like you're holding a pencil. Hopefully you don't hold your pencil like a 4-year-old like this. Hold it properly like this. I won't judge you, but please try to hold it like this. And you're going to take the caps off of your test tubes with your pinky and ring finger and your ring finger and your middle finger. So you'll take one cap off and then the other cap off. You'll never take these caps and put them on the ground because the ground is potentially contaminated. So if you put them back on your test tubes, now your test tubes have contamination. So you always hold them in your hand while you're doing whatever you're doing. You'll then take your needle and you will stick it into the tube that's got the stock culture already in it, take it out, and then you will put it into the blank media that you're putting the bacteria into. Really key thing though, when you hold these test tubes, you don't want to hold them straight up and down. The reason why is because straight up and down allows bacteria to fall into them, either from the vents or from you breathing or your lab partner spitting on them. I don't know, maybe they don't like you. Always hold them at about a 45-degree angle. This minimizes contamination. Key word of warning though, if it is broth, make sure you're not like at a 90-degree angle because then it just spills out. So 45-degree angle, grab the bacteria, put it in the one that you did, put the caps back on in the same order that you took them off on, and then put the test tubes down, flame your loop, and now you can put your loop down. You never want to put your loop down and then pick it back up and stick it into something that's got bacteria in it because we want to make sure it's sterile. Anytime we put something on the ground, we have to consider it no longer sterile. We also never want to put contaminated instruments on the ground without sterilizing it first. So we always sterilize before we put anything on the ground, and we don't want to put anything on the ground that we want to remain sterile. Make sense? Okay. That's done. Next one up. There's my, there we go. Streak plates. So the streak plate is the other type of major media we use. It's commonly used to separate different types of bacteria from a mixed culture. Mixed culture is very, very common in the environment. Most bacteria exist in the world in a mixed culture. Sometimes we want a pure culture, which is only one genus and species. And the way we do that is we get one isolated colony by itself on a streak plate, and we can say that one isolated colony is probably just one bacteria that made that colony. Now, there are many different ways you can make streak plates. So this is one design example that people often use. This is a bacteria, E. coli, with a pGlow gene in it to make it glow. That one's a little bit complicated. This is the one that we use in lab because it's simple and easy to replicate and do a good job on. Okay, and don't worry, I'll let you guys go soon. We're almost done. The way you do this, and I'll show you guys the pattern here in a second, but the way you do this is you always want to lift up the lid just a little bit. You never want to take the lid all the way off, because when you take the lid all the way off, now it's just open to the air and contamination from you breathing or stuff falling in from the sky, and really the ceiling. So always just lift the lid up just enough to do whatever you need to do and put it back down. So you always lift the lid up with your non-dominant hands, and you'll take your looper needle and just kind of stick it in there, and you always want the looper needle to go kind of towards where your palm is, because that gives you the best control when you're doing stuff on here. So for the first one, you will take your plate, you will draw a line down the center and a line across, but you will do this on the back of the plate. You will not do this on the lid, because whenever you label lids, well, I can just take the lid off and put it somewhere else, put it on another plate. And now you don't know what this plate is. So always make sure you label the bottom of the plate, because the bottom of the plate can't be removed from the auger, so it stays with it. You will then hold the plate with your hand, the palm of your hand on your non-dominant hand here. You'll lift the lid up, you'll take your loop, you'll sterilize it, you'll grab the bacteria from your stock culture, and you will move it in this direction towards your palm, and you'll start right here, and you'll do this zigzag pattern, like this. And this is helping spread out the bacteria so that there's a nice even line there. After you guys are done with that, you will close the lid, take your loop, flame it again to sterilize it, take it back out, and you'll turn the plate 90 degrees so your palm is now here. You'll lift the lid up, going in from this direction with the loop, and you will drag across these lines here and streak back and forth here. You are not actually putting more bacteria on this plate, you are just moving the bacteria over. And the reason why is because we're trying to reduce the number of bacteria to get an isolated colony. I'll explain why in just 1 second. Once you do that, you will flame your loop again, sterilize it, get all the bacteria off, turn it another 90 degrees, and you'll do the exact same thing coming over here and dragging the loop back and forth here. Key thing here, you want to avoid going over the top of these arches right here, because these arches actually have a whole bunch of bacteria on them, more than you think, more than the lines by themselves, so just keep that in mind. The reason why we do this is because that first streak that we did on the very first quadrant, it's probably going to be a solid streak. There's going to be no isolated colonies on it whatsoever. So we need to lower the number of bacteria on there, and that's why we flame our loop in between, because we're just moving the bacteria over and trying to reduce the number to get an isolated colony. And when we say we have an isolated colony, we can say that if it's not touching anything else, that's probably just one bacteria of one genus and species. So that gives us an isolated, pure culture for us to work with. And that's a very important thing for the rest of the semester. Okay. So what we'll do on day one is we will give you a TSA slant, a nutrient broth, and a TSA plate. You'll use a Sharpie to label the test tubes and the plates with the organism, your name, date, and section. Specifically on the tubes, what will you label? What part of the tube will you label? The tube, not the cap. Very good. Okay. You'll then transfer E. coli into the slant and Saccharomyces cerevisiae into the broth. And finally, you will make a streak for isolation plate with this bacteria called Serratia marcescens. It creates a nice vibrant red color, so really easy to see. You place all of your cultures in your group's designated rack, and then you will put them away in the designated spot in your incubator. Your TA will tell you which one it is. And then on day 2, we'll work on pipetting. If you guys haven't done pipetting before, it's really easy, but it does require a little bit of practice to kind of get in the swing of things. The basic parts of a pipetter are actually really easy, and they all are almost identical in basic shape, but the ones in the lab are all like this one. The top part is a plunger. That's the part that you press down to suck up the liquid. This is the part where we put on the tip that sucks up the liquid. Right here is the dial that we use to adjust the volume, and this is where we can see what volume we set it in to suck up the liquid appropriately. And lastly, we have the tip ejector button, which moves this little metal guy right here to shoot the tip off once we are done using it and dispose of it properly. Now, each pipetter has a different set of volumes that it works with and a range that works in its kind of middle part the best. Almost all pipetters are named after what kind of volume they do. So there might be a P20 or a P200. Other pipetters will have like a different letter or stuff there. You just have to look at it and it tells you what the maximum volume is. So in this case, a P20, its maximum volume can be 200. A P1000, its maximum volume can be 1,000 microliters. The way you read these are, you start with the very top number, and that's going to be the top number, the first number right here. In this case, these are the P20. The first number is going to be the tens point. The second number is going to be the ones, and anything after this is going to be the decimal. So you could set this to 150, that's 15 microliters, but if you set it to 15, and then you moved each of these tick marks over until you get to the 5 tick mark, that would be 15.5. So that one can do decimals, fractions of a microliter. P200, that means that the first number right here you'll see is in the 100 spot. This one's going to be set to 7, so that's 70 for this guy. And this last number is this guy right here, so that's going to be 75. Now the tick marks that you see here at the bottom, same kind of deal. You can actually set this, so if you click it over 1, this will be 75.1. Now, how close is it to 75.1? Well, not very close, but you can kind of fudge it a little bit, but just know that that's what those tick marks mean. They're, in this case, are working on the decimal part once you get to the tick marks. The last little part here is the P1000. Its maximum volume can go to 1,000. Typically, you don't use it at 1,000 because that's just not super accurate, but that's why it's got a red number right here. So you can only go up to 1,000. You can't go over 1,000 on this guy. So the next number is going to be the hundreds, and then the number after that is going to be the tens, and the tick marks right here are actually the ones part of this guy. So this one is 500, so that's 500 microliters. And that's basically how you do this. Your TA is going to show you an example of how to do these, but if you guys have any questions, make sure you guys ask them. What you guys will do on day 2 is you guys will use the pipette to pipette into 3 separate microtubes, different volumes of dyed liquid. Once you kind of get a hang of that, then you guys will observe your plate and tubes from day 1, and you will make a wet mount slide, adding in 25 microliters from your E. coli, 25 microliters from your Saccharomyces, which is a large fungi, and put that onto a slide, as well as scrapings from your cheek cells. We will give you guys sterile toothpicks to scrape your cheek and just rub it into that liquid that you put on the slide. And lastly, you guys will transfer hair onto the slide, and you guys will be able to bring them all into focus. And you can actually see them all on here. The hair is just really easy to focus on, but because these don't have any dye on them, they're a little bit tricky to bring into focus. Your TA will show you an example of how to do this on the instructor microscope. But other than that, it's pretty simple. Now, before I let you guys go, hold on. If you guys have any questions, come see me after class. We'll talk. Make sure you guys complete all of your safety forms on Lab Archives, also your safety quiz on Canvas. And before you guys come into class on Tuesday at 8 a.m., the quiz 2, the pre-lab quiz, is due. It's due Tuesday at 8 a.m. So you have Monday to do it, or Tuesday morning if you get really early. But just make sure that you are aware it is due early in the morning. I'll see you guys next week. Have a safe weekend, and let me know if you guys have any questions.